Okay, let's get started. Hello, everyone. Welcome to the first Soapbox Science St. John's event. We're very excited to have you here. My name is Sauve. I'm one of the organizers of Soapbox Science here in St. John's, and I'm going to be your host today. But before we talk science, as a settler that lives and works in Newfoundland, I would like to acknowledge that these are unceded lands of the Mi'kmaq and historically of the Beothuk. And also in Labrador, the settlers that live and work there are on the traditional lands of the Inuit of Nunatsiavut and Nunatukavut, and the Innu of Nitassinan. Now, in terms of housekeeping, you'll notice that you were muted and that your camera was off when you joined. We ask that you please keep it that way, because you'll also notice that we're recording. And so for your privacy, we'd like to keep your videos off. But we do encourage you to engage with our speakers and us using the chat function. And so as Christina has posted in there, all your questions can go in the chat, and you can also use some of the buttons that Zoom provides, clapping, hands, maybe yes, no, if the speakers are going to be asking you questions. So Christina and Ozgen, the two other organizers, are going to be monitoring the chat, so they'll be keeping an eye on you. Very shortly, I'm going to introduce our three speakers, and they're each going to talk about their work for about 3 minutes. Then we'll move on to a panel discussion with 2 pre-prepared questions, and then that's when we'll look at audience questions. So you can ask a question at any time, during a talk, between talks, after, and we're going to compile them, and then we'll do our best to get you an answer in the panel segment. So let's get started. I'll tell you a little bit about Soapbox Science first. It was founded in 2011 in the UK to promote women in science and also to bring science to the public. So typically, Soapbox Science events literally put women on soapboxes in public places. Unfortunately, this year, of course, due to COVID, we had to move to an online format, but we're happy to be able to be having the event anyways. So we have 9 wonderful speakers over 3 weeks. And today you'll be meeting Hannah, Ashley, and Wendy. So first, Hannah Wallace is a PhD student in biomedical sciences at MUN, and she's studying to be a virologist. So Hannah has done some work around Ebola, and she's involved in the CAN-HEP-C project. It's a national research project around hepatitis C. And today she'll be talking to us about her work and her talk entitled Viruses and the Cells They Kill. Second, we're going to hear from Ashley Noseworthy. She's the founder and CEO of Edgewise Environmental, which is an environmental consultancy just focused on marine mammal and seabird observation and acoustic monitoring training, which is the first of its kind to offer any kind of dedicated courses on the topic. And the kind of knowledge is really key in mitigating the potential impacts of human-made ocean noise on mammals. And today she's going to teach us how to speak whale. And finally, last but not least, Wendy Lasisi is a PhD student in clinical epidemiology at MUN. She's a recipient of a Rotary Foundation Global Grant Scholarship for multiple sclerosis. And her PhD research focuses on linking the clinical features of MS to brain structures and function and changes in limb impairment. And today she'll be talking to us about MRI, a snapshot of brain damage, and recovery. So without further ado, I'll hand it off to Hannah for our first talk.
Thanks, Sarah. All right, so by now, I'm sure everybody has heard about viruses. They're like a kind of a trend right now. And unless you've been asleep for the last 7 months, then I'm sure you've heard of this guy, COVID-19. So COVID-19 is a disease caused by the SARS-CoV-2 virus, as I'm sure you've heard a lot about. But do you really know what a virus is? Do you know what it does? So today I'm going to take you through the basics of virology, which is the study of viruses, which is what I do. So you probably know that every living thing is made of cells. That's me and you, your dog, and the mosquitoes that bit you during the summer. But cells are microscopic, and viruses are even smaller. And unlike a cell, viruses aren't even considered to be alive, which is crazy considering what they can do. So viruses, I like to call them the little punks of the microscopic world because they can't survive on their own and they get into a host and cause a lot of damage. Now, not all viruses cause damage to their host, but I'm going to focus today on viruses that make you sick. So viruses are, on the most basic level, made of two things. Number one, proteins. This is basically what carries out specific functions. And then two, genetic material, which is basically the blueprint for making more viruses. So you can think of it like this. The proteins are like all of the people working at a construction site. The people who drive the workers to the site, the people who are doing the building, and the people who are overseeing it all. They're all like different kinds of proteins. And then the genetic material is basically the blueprint. So that's everybody at the work site is working with this blueprint to try to make one final product. And so the only job of a virus is to make more copies of itself. But they have a problem. They can't actually do this on their own. So you have to have basically a construction company to help them. And that's where the host comes in. So the virus infects a host, and basically the virus takes over the cell and produces a factory to just make more viruses. And the cell doesn't really like that because the cell wants to make its own proteins. And cells are a little bit dramatic. So if they can't get rid of the virus, they kind of just give up and say, I'm done. And that's called programmed cell death. And so just like humans and animals can die in a variety of ways, cells can also die in a variety of ways. So in my lab, we study one specific form of programmed cell death called pyroptosis. And yes, I know that's a scary word, but I'm going to break it down for you. So pyrop, basically meaning fire, and osis is basically the ending for any kind of cell death. So it basically means fiery death. And this makes sense because what happens in this form of cell death is you have the cell and it explodes. So everything that was inside the cell comes out. And then your immune system, which is basically like the security system of your body, notices that something's going wrong. And so it begins to respond to the exploded cell in a way that can cause more explosions. It's basically like fighting fire with fire. Sometimes it stops it and sometimes it makes it worse. And so our work overall kind of tries to understand how viruses induce pyroptosis and how that might contribute to disease or how that might make you actually sick.
That's super cool. Thanks, Hannah. That's a really great analogy. I'm really surprised to hear that viruses aren't considered to be alive. That baffles me. Anyways, moving on to our second talk. Ashley, go for it.
There we go. I had to be unmuted first. There we go. So welcome everybody. Thank you for stopping by and listening. My name is Ashley Noseworthy, and I am the CEO and founder of a local environmental consultancy called Edgewise Environmental. And my work centers around marine mammals, seabirds, and anthropogenic noise. And anthropogenic is just a really kind of fancy way of saying human-generated or man-made noise. And I'm most interested about noise that's created in the ocean. So I have a great passion for sound, how marine mammals use sound, and how it is regulated across the countries of the world, because it tends to be different no matter where you go. And it's important to understand underwater noise, both naturally and unnaturally made, because it can have various effects on different species of marine mammals. And there's lots of variables when it comes to sound in the water. So, you know, we can go through temperature and pressure and depth, and those might be your top three. So let's start with some really interesting facts about underwater sound. The first being that sound travels approximately 4.4 times faster underwater than it does in air. So it can travel at kind of depending on the frequencies, different depths from what animals, like I said, lots of variables, but 4.4 times faster. The blue whale creates the lowest sounds amongst any marine mammals, as low as 10 to 15 hertz. This is below our human threshold. So as somebody that works in this field, if I have generated sounds or if I have recordings of blue whales and I bring them onto a screen, I can only see those sounds unless I apply filters or I speed it up so that my limitations to human hearing can actually hear some of those sounds created. And there's actually a channel in the ocean called the SOFAR channel. And SOFAR stands for sound fixing and ranging. And that's where sound can travel the fastest. So it's believed some of the large baleen whales, like the blue whale, can actually use this channel to communicate across the globe, which is pretty incredible. So let's focus on the three things that we would need to create sound. And this is sound in air or in water. And this can be from humans, animals, machines, it doesn't matter. We need three things. We need pressure, we need a wave, and we need a medium. We need a way for that sound to transfer through the environment. So for humans, pressure is created in our diaphragm and our lungs. And when we create that pressure, it goes across our vocal cords, and our vocal cords will vibrate. And our medium is air. So if you want to, you can take your fingers and you can press them on your neck and you can just create a sound. And you'll actually feel that vibration. It's nice that we're all muted so we can't hear one another when we do that. For marine mammals, many create sounds in the same way in that they need vibration and their medium is water. But if you were to speak whale, or if you were to speak dolphin, you need some different organs, and they create sounds in some very different ways. So unfortunately, as humans, we can't speak whale, but one of the most important things we can do is listen. And this is where I typically function, is in a field called passive acoustics. So we want to listen and we want to understand, and we can learn a lot about just listening, same as we can with humans. So there's a number of tools scientists can use to record sounds underwater, the most common of them being hydrophones, which is an underwater microphone. And here we can record sounds and analyze them. Not all of them can be heard, similar to how a dog can hear high-pitched sounds, but humans can't. So how does this relate to man-made sound? Well, we can record and listen sounds, and sometimes they can overlap, similar to us being in a concert. The concert can be the same sound as us trying to communicate with our friends. If we're too close, if that concert is kind of the same tones or frequencies as we're speaking, we have to shout. So we either have to get louder or we have to move away from the concert. And that's the same for marine mammals and certain generated sounds. So what is really interesting in the past year with the pandemic is that we've seen a significant reduction in anthropogenic noise in certain areas, which has allowed for scientists to record in areas or potentially hear or observe marine mammals in areas that were difficult or they perhaps weren't seen before. And we call this an anthropause, which is pretty incredible. So I'd like to leave on a note of what you can do. I always like to bring it back to how can a person even not in this field potentially help or do something. As people who live in Newfoundland and Labrador, we tend to spend a lot of time on the water. So if you are on a vessel at any point in time, be sure to keep your eyes out for marine mammals. Remember, you need to stay at least 100 meters away. That is a legal requirement. And that vessel noise may attract or deter marine mammals. So don't bring the concert closer if they want to move away. Then if they want to interact, you just shut down your motor. And that's it.
Awesome. Thanks so much, Ashley. And now we're going to have Wendy give her talk, and Ozgen's going to share her screen for a second so that we have a visual.
All right. Hi, thank you for coming to this talk. My name is Wendy, and I study the brain. Now, as a neuroscientist, I believe that the brain is the most important organ in the body. Just like all the other organs in the body, the brain can get injured or it can get damaged. And in fact, as a person gets older, brain cells die, and they find it increasingly difficult to grow back. You can compare that to when a person gets sick and the sickness they have or the illness they have affects the brain. That normal aging process of brain cells dying and finding it difficult to grow back becomes sped up and their brains start to look like and act like it's way older than it actually is. So when we suspect brain damage, there are two main questions we ask ourselves. And the first question is, where exactly is the damage and how bad is the damage? The second question we ask is that over time and given treatment, does the damage stay the same? Does it get worse, or does the brain recover? Now, to answer all of these questions, we use a tool called MRI, which is my favorite tool ever. MRI stands for magnetic resonance imaging. So it allows us to take pictures of the brain without having to cut open the skull. So I have an image there. The first image, image A, is an image of a slice through a human brain. So when you look at that image, you see that it's an image that is taken of someone that is not living, because to see the brain, the person would have to be dead, and then we cut open the head. But MRI allows us to take pictures of the brain in a living person. So we're able to see images, we're able to take pictures of the brain while a person is still living, which is quite awesome. It uses very big and powerful magnets, radio waves, which are the same waves that our cell phones use, and a very complex computer system. And with all of that, it produces very high quality images of the brain in grayscale. And I have an example of that in image B and image C. So if we look at image B, it's just an MRI of an average brain. Nothing looks out of place. The cells are tightly packed. If you compare that to the last image, which is image C, that is an image of a damaged brain, or a brain with extensive damage. When you look at the edges of the tissue, you can see that there's more space in between the tissue, and that is an indicator of loss of brain cells. If you also look towards the middle of that image, you see some bright dots that appear. Those dots are indicators of inflammation or injury. So at my lab, the Recovery and Performance Lab, as part of our research, we collect and analyze MRI data for 3 reasons. So the first one is to measure the extent of brain damage. The second is to measure how good the therapies that we test are at either helping the brain recover or to slow down the process of damage. And lastly, we take all of that information and we try to see if there's a relationship between how our hands function and how the legs function. So people say that pictures are worth 1,000 words, but I like to say that MRIs of the brain are worth 1,000 words or numbers, depending on how you look at the images. Thank you.
Awesome. Thank you so much for those 3 great talks. We're going to open now for a panel. Please remember, you can ask all our speakers questions through the chat. You can ask them specific questions about their talks, about their work, or about their experience as women in science. And you can direct that to any speaker or to the panel as a whole. But I'm going to kick it off with our 2 pre-prepared questions. So we want to ask all our speakers, if you could change one thing about the scientific culture right now, what would it be? So I'm going to ask Hannah to answer that first.
Okay, this was really hard for me to pick one. So I picked kind of 2, I cheated a little. So one is not publishing negative results. There's just a trend of only publishing positive results, and you can so easily waste so much time trying to figure something out, especially in lab, like in basic science research can take so long, so much time wasted. And if people just publish their negative results, we would get there a lot faster. And then the other thing that I would change is glamorization of like the hustle and working so hard that you like drop. Because yes, you need to work hard. It's, you know, you signed up for it kind of thing, but I don't think it's healthy to glamorize that.
Yeah, for sure. Ashley, add anything?
Hannah, you stole my answer. No, it's interesting that we both have similar answers. So my answer to that question was there is this perception when you are in science or business, but science, that it's all about the hustle and grind, and that if you are not writing, thinking, doing science, that you're not somehow true to the program or to the data or to the science that you're doing. And I think that balance is really important. So I would change that, because balanced, happy, healthy scientists, I think, are the most productive. And so I would change that.
Awesome. Wendy?
What I would change really is how long it takes. So I call it the dissemination process. So how long it takes for the results that we get at the lab to actually be implemented like in real life. Because like, for example, at my lab, we have participants in, we see that an intervention or a therapy that we're using is very beneficial, but we know that once the study's over, the participants aren't going to get it anymore unless it's implemented in healthcare system. So what we try to do is have a focus group and talk to people in the community that suffer from the disease that we investigate, and tell them, oh, look, we found this, you should try this. But obviously there's a problem if we tell them try this, but they don't have the tools to try that. So I think what I would change is how that the results that we get, that we know they work, like how we can get that into the public, and how like healthcare can get on it and start providing this for people.
Yeah, absolutely. If you'll allow me an introduction, that's a problem I think that we have with music and things like music therapy. Like music therapists know that it works. It's not going to cure everything, but like it has a very specific and very positive role. But it's really hard to convince, you know, governments and powers that be that like you need to fund this and that this matters. I also wondered, since you both mentioned or brought up the publication of negative results, if registered reports are something that is starting to be a thing in your fields?
I know it wasn't mentioned, I didn't mention it, but one of the things we struggle with, at least in marine mammal science, is the time that it takes. And so one of the changes that we're starting to see is if there is something that is really unique or something that needs to get out fast to the community, we'll see that as a note or a report in a scientific journal. But normally I'm reading data that is anywhere from 2 to 5 to 10 years old.
Yeah. The idea, this is starting, I know for sure, in psychology. So the idea with a registered report is that you write a paper in your methodology and your analysis before you do the study and submit it for peer review. Then it gets, what's it called? Conditional, I guess, conditionally accepted. And then no matter what the results are, it'll get published, because we've already decided that the science is sound and the question is interesting. And so I think that's an interesting, at least, development, and has potential to help us both stop or slow down the publication bias and speed up the publication process. Because if everything's already pre-accepted, it should, in theory, be much quicker from data collection analysis to publication. And so that's something that I think is really good about psychology, and at least I hope that spreads. It's definitely a problem that we have. Cool. Our second question is, what would be your top recommendation for a woman studying or working towards a career in STEM? I'll go the other way and ask Wendy to answer first.
All right. My top recommendation, I guess I would just say, is it really depends on you and what you love, what your passion is, what you feel like you will be dedicated to. And it's always nice to have like a plan as early as possible. Now, we know life doesn't always work that way. You could start out with one plan, but if you have a general idea of like what you would like to do, like, would you like to be in medicine? Would you like to be in engineering? Whatever it is have a general idea as early as possible. and then always look for opportunities that you can use to explore those ideas. Because when you do get those opportunities, you're able to know, okay, I do like this, or I don't like this, or maybe I should try something else. But if you don't have an idea and test out, then it becomes difficult to get that goal, I guess, is what I would say. So start as early as possible and get as much experience as you can.
Awesome, thanks. Ashley?
My recommendation would be to take advantage of all of the opportunities that are put forth in front of you, even if they don't seem directly related to the plan that Wendy was talking about, which I think is really important, or if they seem like they're on the periphery of your skill set, explore that, because it might lead you in a really interesting direction, and learning that you are not interested in something is equally as important as learning as what you are interested in. I would also recommend looking for mentors and people to look up to and talk to about being in STEM, or specifically a woman in STEM, about somebody who's in that field. And the earlier you start that, you really know kind of what's coming down the pipeline. And so it kind of sets you up for success, or you can mitigate some of those milestones or hurdles that you might face.
Mm-hmm. Hannah?
I have a little bit of the same answer as Ashley again. For me, I kind of said I would recommend surrounding yourself with a really supportive network. So that's friends, family, but also your mentors and supervisors that are going to support you, because as a woman in STEM, there will be times where you won't believe in yourself, but I think it's really important to surround yourself with people that do believe in you. So when you have those hard moments, there's someone to kind of support you. And we know, like, women in science are more accepted now than they used to be, but there's still some fields more than others that's still a barrier. So you're gonna want people like in your corner.
Awesome. Thank you all three for all your advice. We're going to switch now, the last half hour, to questions from the audience. So far we have one, but hopefully they start trickling in. So first from Noel, I assume this would be for Wendy. Noel asks, does the MRI interact with the living brain? As for example, X-rays interact with the body.
Right, that is a very good question. So MRI works in a similar way that X-rays work, but they use two different kinds of waves. So I don't know if you know about the electromagnetic spectrum of like the different waves. So on one end, we have radio waves, you have visible light somewhere in the middle, and then you have gamma rays on the end. So X-rays work with X-rays, like it says in the name, and MRIs work with radio waves, and they interact differently with the body. So how radio waves work with the body is it works with the water that actually flows through our body. So it sends out waves into the body that interacts with hydrogen in our body, and then sends a response back to the computers, and they're able to take all that data to construct like an image of the brain based on where water is in the brain, if that makes sense.
Cool, thanks. And actually, I'm curious about where you are, and I wondered if you could, I'm assuming it's your lab, and if you're in your lab, do you want to give us like a little tour of what's going on there? I don't know if that's possible with the camera. Is that difficult?
Hold on. Okay, I was muted for a little bit. Yeah, I can show you just this little corner of the lab. So unfortunately, we don't have an MRI at the lab. We don't have the money to have a research magnet here. So we use the MRI at the hospital. But in the lab, so apart from MRI, I actually work with other imaging techniques or brain imaging techniques. So right there on the side, is something we call TMS. So that's transcranial magnetic stimulation. And how that works is by sending waves through the head, and then we're able to get waves and analyze that. On the other side right there, I don't know if you can see that, that's another equipment that lets us actually also send waves. So we work with a lot of waves, send waves through like the hand. We can stimulate muscles in the hand, stimulate muscles in the legs, and then we get waves, and you analyze that wave to see what looks off or what looks normal. Yeah, that's my little corner here. But in our lab, we also have a physiotherapist, because that's how we administer our treatment. So the big treatment that we have in the lab is actually aerobic exercise, and we pair that with other things. So yeah, and just right there's, I have MRI pictures of the brain, basically. So I look at a lot of pictures. If I'm not looking at pictures, I'm looking at waves. It's one or the other. Yeah.
Cool, thanks for that. As a follow-up question from Noel, can MRI damage a brain?
Great. MRI is the safest technique that can be used on the body. So it doesn't damage the brain at all. The only safety issues we think about is if someone has metal in their brain, because MRI is a magnet. So you don't want any metal next to that. But it's the safest technique that's actually used for imaging. It's safer than X-rays. Yeah.
Sure. Awesome. Yeah, my permanent brace makes me sad that I can never see a photo of my brain. Because I just got like a piece of metal in my mouth. Not go well. All right, we have a question for Ashley from Jack. They ask, is there much variation between marine mammal species in how they respond to anthropogenic noise? Do you look at seals and other mammals too, or just whales?
That's a great question. So in terms of the variation of how they respond to anthropogenic noise, I'll try and kind of give just a basic, pare down the answer, in that the response is dependent on things like the proximity to the noise, the loudness of the sound, and the frequency that it's emitting. So for example, smaller toothed whales like porpoises or dolphins, beaked whales are on the higher end of the frequency spectrum. And so things like sonar, as well as cavitation from small engines create those higher pitched sounds, which can potentially cause masking or higher rates of effects. And then the lower frequency sounds, think of your large baleen whales, and then think about large vessels. It could be things like seismic or renewable energy. They tend to create lower frequency sounds, which can cause potential effects. Again, it all depends on those variables. I do look at seals, and I have looked at seals previously. One of the reasons why I might have kind of skipped over seals is because seals actually create sound very similarly to how humans create sound. But one of the interesting things about seals is that their way that they create sound might be similar, but they can create different spectrums or frequencies of sound in air compared to water. And so after this talk, if people are interested, you should go to a website. It's called DOSITS, DOSITS.org, Discovery of Sounds in the Sea. And there's an audio gallery on there where you can listen to sounds of seals, dolphins, whales, anthropogenic noise, and what they sound like underwater.
Okay, very cool. We have a question for Hannah from Kelsey. She says, Hannah, you said you were studying pyroptosis. What aspects are you studying? And this is the effects this has on the immune system or the mechanism leading up to cell death?
So I look at several different aspects. And the kind of interesting thing about pyroptosis is that in some cases, it acts as an innate immune response. So your immune system is divided into innate and adaptive immune responses. And your innate happens kind of automatically right away when you encounter a pathogen or something that has invaded your body. So there's that one aspect that this cellular pathway that leads to pyroptosis is already in the cells. So in some cases, the way the cells respond is a mechanism of protection. But in other cases, kind of when pyroptosis begins to happen too much, you can actually have pathogenesis from that. So disease caused by too much pyroptosis. So a really good example of this is influenza viruses. So when they infect your like airway, you need pyroptosis actually in order to get an adaptive immune response. That is one thing that's known about these viruses. You don't get an effective immune response if you block the pyroptosis pathway. But also, if that gets out of control, you can have lung damage, and it can be attributed to that. So I study it with both of those lenses on it and in the context of several different viruses.
I have to mute myself first. Awesome. And I actually have a question also related to your work. How does your field define life? If viruses are not alive, what is alive?
They are not. And there are many criteria, and you could probably argue with anybody in the field for quite a few reasons. So the biggest thing is that they can't survive without a host. They are an obligate intracellular parasite. So, and it's really hard to not anthropomorphize viruses. It's so easy to say like they do this, they do that, but they don't do anything. It just happens. And so in terms of like replication, they have to be inside a cell because they have to use the cell's machinery to make more proteins, more genetic material, and then they can't grow. So it's kind of those two are the biggest parts of why we would say the viruses are not alive. And I think the more important one is that they can't survive on their own. They can, some viruses, particularly protein capsid viruses, so like norovirus that causes like big diarrhea outbreaks on cruise ships, it can live, but it's not living, so it can survive on a surface for a very long time, but it's not reproducing itself, so it's not alive. But that's why you can have really big outbreaks of that, because it can on door handles and things.
Okay. As a follow-up question from Noel, to kill a virus, is soap or alcohol or something else most effective?
Soap, alcohol, both of those definitely kill, some more than others. So you can also classify viruses into two groups based on the outside of them. So some of them are enveloped, so they almost have a membrane, and a lot of them steal that from the cellular membrane. And those ones we call in the field, we call them wimpy viruses because soap and alcohol kill them fairly easily, like influenza or hepatitis C virus. Even SARS-CoV-2 that causes COVID-19 is enveloped, and we would consider that wimpy. But then there's protein capsid bacteria, and because they're encapsulated in a protein, they're a little bit harder to kill, so alcohol is going to be more effective for those kind of viruses, and that would be like poliovirus that is stable in contaminated water for a long time, or hepatitis A, something like that.
Cool, thanks. I have a question for Wendy from Paul. They ask, for people who choose to donate their brains to research at death, how do you conduct your examination, considering you mentioned MRI is usable only on living people?
All right. I mean, the reason why we do have MRI is to be able to measure in a living person so that we could help the living person stay alive. But once a person is dead, then the way that we conduct research in that, so there's clinical research that's only done in living people. And then there's benchside research that we do with dead brains. And to do that, we take slices of the brain and you get staining agents depending on what you're looking for. So that way you actually have the slices in front of you to work with. But obviously it's not living, but there's still a lot of research that you could do with those slices to, you can see how a disease affected a brain, what it did to the brain. So that's how you would deal with a dead brain in comparison to a living brain.
Well, thanks. We have a question for Ashley from Christina. Is anyone conducting noise research in freshwater systems, and how does it compare to ocean noise?
There are, and they could be freshwater or brackish, so a combination of fresh and salt. And a good example of this would be your river dolphins, so Amazon river dolphins, Yatanzi river dolphins. There are different types of marine mammals that can inhabit certain types of water, so it might not be truly freshwater, but those are being looked at in terms of vessel noise up and down those particular rivers and how they're relating to low population numbers of those particular river dolphins. And in freshwater, there's also a lot of acoustic research being done on fish. So fish acoustics is a really interesting area in which I am not an expert, but I've slightly dabbled, and you can get some really interesting results. Fish do use sound and they do make noise.
Oh, super interesting. We have some questions for everyone from Jack. I think the one that I want to hear the answer to most is, when did you first want to be a scientist? I'll ask Ashley to start first.
So I guess I'll consider myself one of those outliers, in that if you asked me when I was 8 what I was going to do, I would tell you marine biologist. So I've had an affinity for the ocean since I was very little. And I've been fortunate enough to know and be able to explore that my entire journey through my life. So I am one of the ones that, when you asked them when they were little, they ended up doing that.
Awesome. Wendy?
I also kind of like, for at least since I can remember, I knew that I always wanted to be a scientist, but I didn't know what kind of science. Because at one point I was into space, at another point I was into computers. But I knew that I wanted to do science. Like, I wanted to research. I wanted to like get a question and find an answer to the question. So I knew that I wanted to do science. That's that. But as I got older, I decided, okay, I think I really like the healthcare field. So I think I'm going to go into the medical field. And at one point I was like, okay, I want to be a medical doctor, but I want to be a scientist at the same time. But I guess along the line, being a scientist took over, because I was more into research. Like, I want to do the research. I want to be in a hospital, but I want to do research. So that's, I guess, yeah. But I always knew I wanted to do science. Like, I want to be a scientist, and that's where we are now.
Awesome. Hannah?
From age about 6 to probably 13, you could check that with my parents who are on this call. I wanted to be a paleontologist. I loved dinosaurs. I thought they were the coolest thing ever. And then I realized I was like gonna have to dig in the dirt, and that suddenly became less appealing to me. So I took a brief hiatus from my wanting to be a scientist and thought I was going to be on Broadway for a short time. And then I kind of accidentally fell back into biology, took grade 11 biology in high school and did a dissection, and was like, okay, wait, maybe we can reevaluate this because I really like this. And then I fell into virology because of a very special professor of mine in undergrad. So when I was in my first year of biology was the beginning of the Ebola outbreak in West Africa that ended up being the largest Ebola outbreak to date. And my prof, Barb Dowding, actually incorporated that into our classes and kind of taught us how we didn't have vaccines, we didn't have antivirals at that time, which now we do, which is fantastic. And that was like, I was hooked with viruses from then on.
Awesome. I'm curious now, as like a follow-up question, what do you think made you want to become a scientist from such a young age? Like, that you were so sure, right? Like, were there role models that were involved? Like, what do you think was that about?
This is a good question also. I guess, so for me, I don't remember exactly what triggered the dinosaurs. It might have been my older cousin who, because he always talked to me about dinosaurs and like would tell me about them. But I also attribute that to me like wanting to do science or whatever to how I was brought up, because my parents always said I could do whatever I wanted. And I remember for a while being like, I'm going to be a part-time scientist and part-time ballerina when I was like 5. And they were like, cool, you do it. Like, and I always had people around me that really supported. So I think for me, that was kind of what triggered.
Awesome. Wendy, what about you?
Okay. For me, when I was younger, I really can't remember because it's always been like that. But I think maybe I was just really interested, like when I came to school, in the sciences, like biology and chemistry. I wasn't a fan of physics, but that's haunting me right now. But yeah, so I was always into chemistry and biology. And for some odd reason, I actually did the worst in those subjects. But maybe because I did so bad in them, that's what made me like interested in them. Like I want to do better. But in relation to like being a neuroscientist, I remember the day that I decided that this is what I want to do. It was a biology class, and we were our homework was to draw a human cell, an animal cell. And there were 5 of us in class. Everyone went home and drew the normal standard animal cell, and I drew nerve cell just because I thought, oh my god, this cell is amazing, it looks so different. And after that day, I was just interested in the brain. Like it was so bad that whenever my mom would like go to buy like meat, I would be like, please bring a piece of the brain back for me. Like I really want to check that out. And that's really how I got into neuroscience. And like this is the best organ in the body, I got to study this. Yeah.
That's super cool. Ashley, what about you?
I think for me, I mean, I think there was a few kind of pivotal moments. When I was really young, I had always had a fascination with the ocean, and this is going to be a bit of a throwback, but I distinctly remember I was on like a really old school computer now, and I was on this program called Encarta, and I don't know if people remember that, but it was like an old school encyclopedia, and I was just looking up everything on killer whales, everything I could find. I was printing it off and I was highlighting it, and I was just reading everything, and I was buying books, and so I always knew like marine mammalogy was the field I wanted to be in, and just an unnatural obsession with marine mammals. But when it comes to acoustics, similarly to say like Wendy in physics, I mean, I never did physics in high school. It was always very targeted at biology and some chemistry. But I had gone to Costa Rica on a volunteer project because I was having kind of one of those pivotal moments in year 2 of biology where it's like, do I want to do biology or do I want to do something else, which had never happened. So I went on this program, and we were studying humpback whales and pantropical spotted dolphins in an area next to Panama in the Gulf of Dulce region. And the professor that I was working with actually put me on a separate project, and this is where I was introduced to acoustics. And he said, here, analyze this, I need you to do this, this, and this. And I sat down, and this was my jam. This was what I was meant to do. And I was fascinated and obsessed ever since.
Awesome. Here's another question for everyone from Kelsey. What is everyone's favorite part of their field or of being a scientist? Hannah, do you want to start with this one?
Okay. Oh gosh, this is really hard though. I think that this is almost gonna sound crazy, but my favorite thing that I get to do, and it's not even that exciting of a thing, but it makes me excited, is like when you take out a vial that's like a whole, like a mill, and you know that there are millions of virus particles in that vial. And I just think that that is like the coolest thing ever. And every time I get to work on a new virus, I'm like, look at, like, it's in here. Anyway, that's my favorite thing, I think.
Awesome. Love the small pleasures, right? Wendy, what's your favorite part of being in scientist or your field?
My favorite part is getting to work with patients. Like that is just the joy in it. Just having patients come in, you see they have a problem, and you're trying to help them like be better and get better. And also seeing that sometimes what you do recommend is actually working, and they're coming back and telling you, yes, this is working. That's my favorite part of doing what I do, really.
Awesome. Ashley?
My favorite part would definitely be being in the field, on a boat, on the ocean. You can't see the shoreline. I've spent years of my life offshore. That would be the best part of, figuring out and troubleshooting equipment, of realizing that you have a detection in real time, but you can't see them on the surface, but you can hear them, and then trying to figure out where they are and what they're doing. So kind of being a detective while rolling around on a boat. And I just have to add, it's to the point where I mentioned kind of like discover and figure out what you really love. Like when Hannah mentioned that little vial and millions of bacteria, I remember sitting in microbiology for literally 15 minutes and walked out. It was not my field. And so it's really important to have people that love this kind of stuff, because it's so important, and for other people to recognize that they are good at other things.
Yeah, for sure. I think that's one of the really important things, and especially about bringing women in science and bringing more diversity in science, is that we all bring a different point of view, and we need all of those skills and all those different points of view to work together to really be able to move forward. Okay, we're getting to the end. I do want to squeeze in this other question, and we'll see how much time we have after. Jack is asking everybody, what is a hurdle you faced during research, and how did you overcome it? Wendy, do you want to take this one first?
Okay, I guess the biggest hurdle for me is when you've planned out what your project is going to be and COVID happens and you're not able to do what your whole project was supposed to be, and you have to, well, you do have to write your dissertation, so you have to then do something else. And why that would be really difficult is, for your project, you spent like 2 to 3 years getting the training and planning for that project that you're going to do. And then you have to switch it up because, well, right now it's because of COVID. It could be because of anything else. And it's almost like, well, all that training you got before, it's not that it's useless, but it's not useful at that particular moment. And you have to scramble and get everything together to do things on time and get your research done on time. That's the biggest one for me.
Yeah, I have several friends that I know who are either doing their PhDs or in research where like COVID just totally derailed everything. But yeah, we adapt, we continue. All right, Hannah, how about you?
I think for me, maybe I'm a bit of an anomaly in science, but I will fully admit, I really wear my heart on my sleeve. And I don't like the analogy of like you have to have a tough skin. I don't think that science is going to grow by having everybody that has a tough skin in science. And you need to be passionate about what you do, because I think that also produces really good scientists. But I have had to like accept some criticism, and that's fine. I've also had to step up to some people that have said some not super nice or supportive things, and as a person who wears their heart on their sleeve, that's a little difficult.
Yeah, thanks for sharing that. Ashley, what about you? What's a hurdle that you overcome?
I think, I guess like a hard hurdle, I'll call it like a hard hurdle and a soft hurdle. One of the hard hurdles would be kind of relating to Wendy, the expectation of, for example, when I was completing my master's, we actually lost a piece of equipment. And so I had 2 acoustic devices at one site and 2 acoustic devices at another. And when you go out and you're putting things in the ocean, you understand the risks. But when you go and one has drifted away into oblivion, all of a sudden your data is difficult to be compared. And so it's just that expectation that can be a big hurdle to get around, but that's use statistics and hopefully modeling. I would say a soft hurdle that I regularly have to overcome, or perhaps that I've come across fairly regularly, is being underestimated, which I consider a bit of a superpower. So if I'm underestimated, what will tend to happen is I'll get on site, either because of my gender or my age or something like that. Sometimes you can just tell that they're not assuming that perhaps you know as much as you do. And then all of a sudden when you start to exude some of that expertise, sometimes you can see the head nods and things like that. So overestimation or underestimation can certainly be something to overcome, but I view it and I try and view it in a positive spin.
That's great. Thanks for sharing that. And we're coming close to 6 o'clock, so we're going to have to wrap it up here. I want to say a big thank you to all our speakers for sharing all of their knowledge, expertise, and experience as women in science. And thank you for being our first 3 speakers. I'm sorry we didn't get to all the questions that were posed. I'm going to ask, here we go, a couple seconds. Does any of the speakers have anything else they would like to share before we wrap up? Any last thoughts? Okay, so I'm going to wrap it up there. I want to say a huge thank you, of course, to all our speakers, to everyone for coming. Thank you to Soapbox Science, based in the UK, for supporting us in helping design this event. Thank you to the Wildlife Evolutionary Ecology Lab for donating their Zoom account in kind. We also do want to give a shout out to the St. John's Farmers Market, who were very open and very excited about having us host Soapbox Science there if we're allowed to be there in person. And so we're hoping that for next year, we will be able to have it at the farmers market. And on that note, I'd like to mention that our next 2 events, they are next Tuesday, the 29th at 2 p.m. And then October 6th is our rescheduling of last week's event. So October 6th at 1 p.m. And then we're also going to be looking for organizers for Soapbox Science St. John's next year. So if you're interested in organizing, please do get in touch with us. Everyone who's registered should have my email address, so you can get in touch directly with me. And I'm going to end it right there. So thank you, everybody, and have a good evening.